Dr. Ashison here, and we're talking about Chapter 3, the visual brain, and Stephen Yantis' sensation and perception. We are on the second section talking about the primary visual cortex. So, the primary visual cortex, or also called area V1, is the first step in the cortex. So remember, the information comes into the eye, it goes through the LGN and all those great places, and the first stop it stops in the cortex is the primary visual cortex. And again, that's area V1. And remember back when we talked about the brain in chapter 1, I said put your hands on the back of your head and you have your hands on your occipital lobe. V1 is in your occipital lobe. Here you can see it's kind of the little yellow areas in the bottom of that pink, okay? And other areas of the cortex are involved as well, but those are further down the path. So again, the first stop on this kind of vision train in the cortex is the primary visual cortex in the occipital lobe. Again, other parts of the brain will get there. It'll get there, whether it be the occipital lobe or the temporal lobe or the parietal lobe. But the first stop is the primary visual cortex. So a little bit about area V1. It's about 2 millimeters thick, and it covers only a few square inches of the brain. And again, it's that first stop. It's the first place it's getting this information about vision. It's very, very highly organized. We're going to only go into a little bit about this organization because, again, your textbook is written for a wide range of levels. So we're not going to go to as in-depth as your textbook goes. Your textbook will talk about columns for ocular dominance and orientation. We're not going to really get into those columns and ocular dominance. We are going to talk about how it's organized in a similar pattern as the retina is, and we're also going to talk about cortical magnification. So these will be things that we'll talk about later on in this video. There's various cells found in the V1 too, as you would guess. There's simple and complex cells, end-stop cells. Again, we're not going to go into the details about those kinds of cells, but it's important to know that there are a variety of things, types of cells in that area V1, and each cell best responds to more complicated stimuli. So if you're a complex cell, you're going to be more handling complex stimuli than if you're a simple cell. Okay, so the primary visual cortex, the first systematic studies of those were done by Hubel and Wiesel, and here they are pictured. They actually won in 1981 the Nobel Prize for this work. And what they did is they found that cells in V1 are most effective by being stimulated by either bars or edges, really narrow ranges of orientation. So think like 45 degrees versus 90 degrees. A cell is going to be like, hey, I really like 45 degrees, or no, I really like 90 degrees. And again, Hubel and Wiesel found this out. And they did this by doing single cell recordings. So they would put an electrode on a single cell in an animal brain, clearly not a human brain, but in an animal brain. They did a lot of work with cats, some with rats, but they did a lot with cats. And there's actually, and they again found this information about the simple and the complex cells. There's a launchpad assignment that will walk you through this, how different kinds of cells respond to these different angles, and how this corresponds to those receptive fields, okay? So it's going to walk you through that, and it does a really good job. And here you can see that we're getting a strong response out of this neuron. Let's pretend that this neuron is this blue cone here, and we get this strong response out of that when that bar of light matches up to its receptive field, whereas we get a weaker response where that bar of light is adjacent or off-center to its receptive field. But again, do the Launchpad assignment that walks you through this. It's very, very helpful, which is why I assigned it. Okay. Other stuff with the primary visual cortex. Again, these individual simple cells, the information that we get from them, and again, you'll do this Launchpad assignment, which will show you that we can figure out whether this cell is activated or it's not activated. But the problem is that doesn't give us enough information so that we can really 100% determine the orientation of some sort of stimulus, okay? So it's not as one-to-one as it seems. It doesn't give us enough information that we can say, oh, this is 100% what's happening. Because as you'll see, that contrast will play a role as well. So if things are dim or if things are light, those kinds of things will affect the firing rate as well. And because of this, it becomes hard to determine orientation 100% of the time. we know that generally the greater the contrast, the stronger the response. So if you have a really, really bright light, you're going to get a stronger response than if you're getting a dim bar of light. And we also know that any small area on the retina contains a bunch of receptive fields, and it has many simple cells covering that full range of orientations. So again, it's hard to say which one is specifically going on here, but there are some ways to kind of start to guess at it. And again, there is a Launchpad assignment about this. So if you look at this graph here, this is what you'll be doing in the Launchpad assignment. You can see that there are different angles of orientation, the bar orientation across the x-axis there. And then you see the response rate, the firing rate, the spikes per second on the y-axis. And you see that you can have an orientation, a response rate for a high contrast bar versus a low contrast bar. And so what you'll do is you'll say, okay, well, if you're getting 40 spikes per second, and I'm showing you a 67-degree angle bar. Is that a high contrast or is that a low contrast? And in this case, it would be high contrast because where 40 intersects with 67 is on that high contrast bar. So again, it's going to walk you through that and allows you to make mistakes and fix them and play and kind of teach yourself this. So again, it's a great exercise that can really walk you through this orientation, turning curves and simple cells, and how both contrast and angle will affect the firing rates of these cells. Okay, so renotopic mapping is the idea, and again, this is one of the ways that the primary visual cortex is organized. So we have various receptive field locations, and they are adjacent to locations in the cortex. So like we talked about in the first video, we said if they're neighbors in the retina, they're neighbors in the cortex. And so again, this is what we're talking about when we're talking about renotopic mapping. So this can be done in various different ways. It can be done in a column, which I'll show you an example of, and it can also be done kind of adjacent. But you'll see that this idea of things are mapped in similar ways on the cortex as they are in the retina. So the renotopic map in the visual cortex is constructed on polar coordinates. So kind of if you think about the globe, those are polar coordinates, okay? So we can kind of have the distance from the center, and we can also have the angle above or below the fovea in the center. So it's being constructed both on eccentricity, which is that distance from the center of the fovea. Remember, the fovea is your focal point. So when you are looking at something, you are foveating on it and you're putting that image on your fovea. Your fovea is, again, that highly dense center of photoreceptors in the center of your retina. So again, this is how far is that from that really strong center. And then the polar angle is, again, the angle above or below in a circle around this fovea. So again, the fovea would be kind of like the core of the Earth, and it's kind of around that. Here we have an image that has two different kinds of visual stimuli. We have the checkerboard circle, and that's the eccentricity map, okay? So how far it is from the distance of the center. And we can see the farther it is from kind of the center of V1. So the stuff that's closest to the fovea in this image is that purple color, and you see that purple color on picture A is closest to area V1, and we go further out in that occipital cortex as we kind of go down the colors pink and yellow and green and blue. So again, we kind of go further out in the same way that we have that going further out in our retina. It's going further out in our visual cortex. The same is true for the polar angle. Here they gave us a kind of a wedge, a slice of pizza shape checkerboard, and it rotates. And again, it's the same idea, is how far away it is. And again, that image is there. Another way to think about this is, again, those receptive fields. If those receptive fields are close together, then those neurons, those cells in the cortex are going to be close together. So in image A, we have a retinal image, and it's different. The different ovals are different receptive fields of various neurons, and they're overlapping a lot. They're all kind of centered in on George Washington's head here. And so what we see is these neurons are actually organized in a column because they are all overlapping that way, and they're all focused in on George Washington's head. We see that those neurons are actually going to be in a cortical column, and they're going to be on top of each other. Whereas we see when those are more spread out, we're looking at George Washington's face. Again, that's our fixation point. In image B, those receptive fields go more towards the periphery. So we're looking at George Washington, but we see these other founding fathers as well. And those neurons are more spread out. They're still neighbors, just as these receptive fields are neighbors, but they're not on top of each other, just as the receptive fields that are on top of each other, the neurons are mostly on top of each other. The receptive fields that are next to each other, their neurons are next to each other. So again, we have this idea of neighboring neurons based on neighboring photoreceptors and neighboring receptive fields. This is also important to think about when we think about this difference of whether it's on the fovea or whether it's more towards the periphery. We'll see this really exemplified in what we call cortical magnification. And so this is the idea that the cortex is not a one-to-one situation. Okay, so when we talked about how different photoreceptors are connected into different retinal ganglion cells, sometimes you have one neuron's connected to one photoreceptors connected to one RGC cell. Sometimes you have several photoreceptors connected to one RGC cell. And what this does is it says certain visual fields are more important than other visual fields. And these are the visual fields that are close to your fovea. Those are more important visual fields than the visual fields that are far away from or more in the periphery. And so what we see is the amount of cortical territory, the part of the cortex that's dedicated to that fovea, is much more than the amount of cortex that's dedicated to the periphery. It's more important for you to have more brain power focused on what you're looking at than it is to have a lot of brain power focused on the things on the side. You still want your brain processing those things. You still want that processing going on, but again, it's that difference. And this brings us back to the idea of convergence. Remember, convergence was that idea. When there's no convergence, that's one photoreceptor hooked up to one RGC cell. When there is convergence, that's multiple photoreceptors connected to one RGC cell. And again, this will give us some of this cortical magnification. So here's a picture of that. What you see here with all the different colored dots, we have the orange on the left. You see the visual field. Our thin-skinned individual here with his translucent skull is looking at an orange dot. Okay, so that's on his fovea. That's where he's focusing. The yellow and the green are closer to it, but by the time we get to pink, we're way, way off in the periphery. So what that's going to look like in terms of cortical space is something like what you see on the right here, where we have a lot more, a lot more dedication to that orange dot than we do to those pink dots, okay? A lot more space is taken up in the cortex for that orange dot than it would be for the pink. So again, this is that renotopic map. Again, it has that same pattern, right? The orange is by the yellow is by the green, the blue is by the purple is by the pink. We see that same pattern that we saw. We see that renotopic mapping, but what we see is the stuff that's on the fovea is kind of magnified. That's what cortical magnification is coming from. It's magnified in the cortex because it's more important to us because we're focusing on it. So remember, this is that idea that fovea really has a high density of these RGC cells. And again, these have really small receptive fields, okay? They're like little bitty compared to the stuff in the periphery. And so again, we have less RGCs in our periphery. And as further away you get from the fovea, the less RGCs we have. And so we have larger receptive fields. Again, this is about convergence, okay? In the fovea, we have usually no convergence. In the periphery, we'll have moderate to high convergence, where we're going to have a lot of convergence in the periphery, whereas on the fovea, generally we have no convergence. Again, we have very small receptive fields in our foveas. So those foveal RGCs have many, many V1 cells to fully cover that area of the fovea. Because we have very small receptive fields, we have to have a lot of brain power to account for each of these receptive fields. Whereas in the periphery, because these are much larger and they have less RGCs dedicated to them, we have fewer V1Cs that are needed for the area in the periphery of the retina. So again, we still see that same mapping where the retina is space-wise, is set up a lot space-wise. Those receptive fields in the retina are set up like the V1 cells in the cortex, but we again are kind of magnifying the stuff that's on the fovea because that's what we're most interested in. Okay, so that ends video 2. I will see you shortly in video 3.